Neuroglia, Chapter 138. Definition: Neuroglia or glia, glia equals glue, is the supporting cell of the nervous system. Neuroglial cells are non-excitable and do not transmit nerve impulse, action potential. So, these cells are also called non-neural cells or glial cells. When compared to the number of neurons, the number of glial cells is 10 to 15 times greater. Neuroglial cells play an important role in the reaction of nerve during infection. Most commonly, neuroglial cells constitute the site of tumors in nervous system. Classification of neuroglial cells. Neuroglial cells are distributed in central nervous system, CNS, as well as peripheral nervous system, PNs. Accordingly, the neuroglial cells are classified into two types: central neuroglial cells, peripheral neuroglial cells, central neuroglial cells. Neuroglial cells in CNS are of three types: 1. Astrocytes, 2. Microglia, 3. Oligodendrocytes. Astrocytes. Astrocytes are star-shaped neuroglial cells present in all the parts of the brain, Fig. 138.1. Two types of astrocytes are found in human brain: fibrous astrocytes, protoplasmic astrocytes. Fibrous astrocytes. Fibrous astrocytes occupy mainly the white matter. Few fibrous astrocytes are seen in gray matter also. The processes of these cells cover the nerve cells and synapses. This type of astrocytes play an important role in the formation of blood-brain barrier by sending processes to the blood vessels of brain, particularly the capillaries, forming tight junction with capillary membrane. Tight junction in turn forms the blood-brain barrier. Protoplasmic astrocytes. Protoplasmic astrocytes are present mainly in gray matter. The processes of neuroglia run between nerve cell. Functions of astrocytes. Astrocytes twist around the nerve cells and form the supporting network in brain and spinal cord. Form the blood-brain barrier and thereby regulate the entry of substances from blood into brain tissues, Chapter 163. Maintain the chemical environment of ECF around CNS neurons. Provide calcium and potassium and regulate neurotransmitter level in synapses. Regulate recycling of neurotransmitter during synaptic transmission. Microglia. Microglia are the smallest neuroglial cells. These cells are derived from monocytes and enter the tissues of nervous system from blood. These phagocytic cells migrate to the site of infection or injury and are often called the macrophages of CNS. Functions of microglia. Microglia, I engulf and destroy the microorganisms and cellular debris by means of phagocytosis. 2. Migrate to the injured or infected area of CNS and act as miniature macrophages. Oligodendrocytes. Oligodendrocytes are the neuroglial cells, which produce a myelin sheath around the nerve fibers in CNS. Oligodendrocytes are also called oligodendroglia. Oligodendrocytes have only few processes, which are short. Functions of oligodendrocytes. Oligodendrocytes provide myelination around the nerve fibers in CNS where Schwann cells are absent. Provide support to the CNS neurons by forming a semi-stiff connective tissue between the neurons. Peripheral neuroglial cells. Neuroglial cells in PNs are of two types: Schwann cells, satellite cells. Schwann cells. Schwann cells are the major glial cells in PNs, refer to Chapter 134. Functions of Schwann cells. Schwann cells provide myelination, insulation, around the nerve fibers in PNs. Play important role in nerve regeneration, Chapter 137. Remove cellular debris during regeneration by their phagocytic activity. Satellite cells. Satellite cells are the glial cells present on the exterior surface of PN's neurons. Functions of satellite cells. Satellite cells provide physical support to the PN's neurons. Help in regulation of chemical environment of ECF around the PN's neurons.